Next, we have Caroline Burns, who's an associate professor at MGH and has been using the Zebrafish model, which we've heard a little bit about already, to understand not only cardiac development, but the remarkable ability of zebrafish to regenerate a heart. Caroline, thanks so much.
All right, so I'd like to thank the organizers for the opportunity to give you a first look into our lab's research program, which is really aimed at using the zebrafish as a model organism to understand and harness the endogenous mechanisms that are governing heart regeneration. Shown here are adult zebrafish. They grow to about 2.5 inches in length, so they're relatively small, and they're very amenable to robust genetics and also to embryonic manipulation because they develop outside of the mom. Importantly, most of the organs within their body will also regenerate completely after traumatic injury. So really the holy grail of cardiovascular regenerative medicine is to come up with new and innovative therapies that will coax the human heart away from scarring and towards regenerating a new wall of healthy and functional muscle. And we believe that the zebrafish actually provides us with the perfect roadmap in order to achieve therapeutic heart regeneration. So the zebrafish heart is roughly equivalent to about half of the human heart. It has a single ventricular chamber and a single atrial chamber as shown here in section. And in 2001, it was published that resection using a pair of simple iridectomy scissors of up to about 20% of the ventricular apex will initially result in fibrin clot formation, so much as seen after an MI in humans. However, very different from humans that will then scar, the zebrafish will completely supplant the fibrin clot with a brand new wall of electrically coupled muscle. Not only are the zebrafish hearts highly regenerative, but also those of postnatal day 1 mice. However, this amazing regenerative capacity is completely lost just 7 short days later. And this really then begs the question of why are some animals so efficiently able to regenerate their hearts while others scar? And we think that we can start to answer this question by drilling down on the similarities that exist between highly regenerative organisms such as these, and then comparing their similarities with non-regenerative hearts and looking for differences. So the field first really wanted to know, how does heart regeneration occur in these naturally regenerative organisms? And the most important question was, where are the new cardiomyocytes that make up the regenerate coming from? And unequivocal data from Cree-Lox-P genetic lineage tracing is shown in both zebrafish and these postnatal day 1 mice that the new cardiomyocytes come by proliferation of the spared cardiomyocytes with relatively little, if any, input from a stem cell population. So our lab and others are now really focused on understanding how is cardiomyocyte proliferation induced following injury, and as a counterpoint, what are the barriers that might exist in higher organisms? So in particular, I'm going to take you through 3 approaches that our lab is currently using to address this question. First, we're trying to identify genetic pathways that promote myocardial proliferation. We're also trying to discover whether certain chromatin states might favor myocardial proliferation over quiescence. And we're also interested in identifying the physical properties of the cardiomyocytes themselves that might confer a regenerative advantage. In regards to genetic factors, we've taken both candidate and unbiased approaches. In a candidate approach, we discovered that the Notch1B receptor is highly upregulated within the endocardial cells, as shown here, of an amputated zebrafish heart as compared to uninjured controls. And in zebrafish, we can use genetic tools in order to suppress Notch signaling over that regenerative window to learn if it's actually important for heart regeneration. And what we found is that unlike control animals that effectively regenerate new muscle that's tropomyosin positive, as marked by the asterisk, Notch suppressed hearts fail to do this. And in follow-up approaches, we learned that this regenerative failure is caused by dampened cardiomyocyte proliferation. And this really then led to our model, which is that notch signaling is activated in the endocardium, which then activates expression of a secreted factor that exits the cell and induces adjacent cardiomyocytes to enter the cell cycle. And in new follow-up studies that are as of yet unpublished, we've discovered that this factor X, or these secreted molecules, are actually Wnt antagonists. In unbiased approaches, what we've done is to fact sort quiescent cardiomyocytes and highly proliferative cardiomyocytes and compare them by RNA sequencing. And what we learned is that there are many genes, of course, that are up and down regulated, and we can then put these genes into categories or Go terms that help us to understand what are the large categories of genes that are changing as cells transition. And what we want to do is then compare these regenerative programs or these high proliferation programs between regenerative and non-regenerative hearts. So the chromatin state configuration may also play a role in deciding whether a myocyte's able to proliferate or not. You can imagine that if the chromatin is open, this would welcome a transcription factor to come in and to activate pro-proliferation programs, whereas if it's closed, it would not be able to do that. So our lab has looked at two different histone modifications that are known to open and close chromatin. One is the K4 trimethyl activating mark on histone 3, and the other is the K27 repressive mark. And to summarize what we've learned, we were very surprised that dynamics within these two marks really are not associated with transcriptional activation. But what we did find is that loss of the K4 activating mark and gain of new K27 trimethyl marks is highly associated with transcriptional repression on a genome-wide level, and this is mediated by upregulation of a histone methylase called EZH2, which is part of the PRC complex. And in zebrafish, we don't have to be satisfied with mere associations. We can actually go in and use genetics to perturb the addition of these new K27 marks and ask, well, is this even important or required for heart regeneration? And what we found is that unlike controls that effectively regenerate, as again marked by the asterisks, these hearts that are unable to accept new K27 marks fail to regenerate new muscle. And lastly, I want to talk about the physical properties of proliferative cardiomyocytes themselves. So one thing that we've noticed a few years ago is that animals that are highly regenerative, their hearts are mainly, if not exclusively, comprised of cardiomyocytes that are mononuclear in nature, and they have a 2C DNA ploidy, or they're diploid. Whereas non-regenerative animals, such as adult rodents and also humans, have polyploid cardiomyocytes that comprise most of their hearts, either binucleated cardiomyocytes, or in the case of humans, around 90% are mononuclear and polyploid. And so what we wanted to do is ask, in a highly regenerative animal like the zebrafish, is polyploidy really a barrier to heart regeneration? And so a talented fellow in our lab humanized the zebrafish heart by making part of the heart now polyploid, with the remainder staying in this mononuclear diploid state. And what he learned was that when about half of the zebrafish heart contains polyploid cardiomyocytes, especially if they are close to the wound region, what he found is these hearts now fail to renew their muscle, and instead they deposit a large collagen-rich scar. And what this then really tells us is that our studies going forward really need to focus on this relatively rare mononuclear and diploid cardiomyocyte population within the human heart, and see if we can coax those cells towards proliferation to confer a more macroscopic regenerative state. So for the next phase of our program, what I've hopefully convinced you is that zebrafish is a very powerful platform for not only identifying new pathways that can promote or govern cardiomyocyte proliferation, but it's also very powerful for assessing the function of already identified pathways or compounds that might promote heart regeneration. And very specifically, what we want to do is test small molecules, maybe partner with others that have found small molecules that drive proliferation in a dish, and ask whether these are relevant in a whole organism setting. We are also interested in learning whether the molecular pathways that we've identified in the zebrafish are sufficient now to promote proliferation in preclinical models. We want to test whether the chromatin landscape is in fact different between regenerative versus non-regenerative cardiomyocytes, with the hopes of identifying enhancers or super enhancers that when open are sufficient to drive proliferation, and lastly, to really focus our efforts on this relatively rare mononuclear diploid population, because we now know that even in a highly regenerative environment, the polyploid cells are failing to divide. So thanks very much for your time.